I'm going to give you a summary of our work to date on neutralizing monoclonal antibodies to SARS-CoV-2. I presented these results previously. I just want to remind you that we looked at a number of patients with severe COVID infection, hospitalized in the ICU, most of whom were on ventilators, and found that the severe patients on the top, as opposed to the non-severe cases, had a more robust antibody response to the nucleocapsid protein as well as to the spike protein, and then greater cross-reactivity to the SARS-1 spike protein. And this difference observed is statistically significant. And associated with this is a likewise more robust virus-neutralizing antibody response. Here, once again, for the severe cases on the top and the non-severe cases on the bottom, with neutralization against the pseudovirus in these panels and against the authentic live virus in these panels. And one could see the curves shifted further to the left, indicating greater potency. And once again, the differences between the two groups are substantially statistically significant. So naturally, when we want to isolate monoclonal antibodies, we focus on the cases that had the most robust response, as measured in this case by the pseudovirus neutralization. And we selected these 5 cases. They're color-coded as shown here. And in all the subsequent slides, the antibodies derived from each of these cases are color-coded the same way. As mentioned, all these 5 cases are individuals who were hospitalized and taken care of in the intensive care unit. Their ages ranged from 50 to 71. They were admitted and were all on mechanical ventilation. That's what MV stands for. And the antibody isolation effort was focused on blood samples obtained from day 18 to day 32. All had acute respiratory distress syndrome. 4 were discharged after prolonged hospitalization. 1 died in the intensive care unit. And what we had done was to take blood from each of the cases, isolate mononuclear cells, and then went through a cell sorting process focusing on single cells that are alive, that are CD3 negative, CD19 positive for B cell markers, and then CD27 for memory marker, and then cells that specifically bound the trimer that was provided by Jason McClellan. And then the trimer, these panels here are blown up so you could see the B cell numbers a little better. You could see in cases 1, 2, 4, and 5, we got substantial number of trimer-bound memory B cells. Case 3 yielded very few. And then these memory B cells were hashtagged and then put into 10x genomics, and single-cell RNA sequencing was done to get the pair heavy light chain sequences. And from there, the antibodies were constructed. So in total, we constructed 252 monoclonals in an IgG1 backbone, and then did transient transfections to get antibody in supernatant to test for antigen binding as well as for virus neutralization. And this is sort of an overview of our screening results. Each patient is color-coded, so patient 1, 2, 3, 4, 5. As expected, patient 3 yielded very few antibodies, so essentially we're looking at results from 1, 2, 4, and 5. And of the 252 monoclonals, you could see many did not bind the trimer at all, but about half of the antibodies bound the trimer. And then looking at the binding to the receptor binding domain of the spike protein, you could see about a third of those that bound the trimer also recognized receptor binding domain. Interestingly, in patient 5 here, of all the trimer-specific antibody, none bound the receptor binding domain. In terms of virus neutralization, you could see these are the positive ones picked up in the screen. In total, there were 61 that were picked up in the pseudovirus neutralization assay, and 41 that were picked up in the authentic virus neutralization. And those with the vertical lines indicate those antibodies that became of interest later on in the characterization. And so here's a table summarizing the results for each of the cases, but I'll just mention the total once again was 252, about half bound the trimer, and of these, a third bound RBD, 2/3 bound regions outside of the RBD. And as I mentioned, 61 neutralized pseudovirus and 41 neutralized the authentic virus. In terms of the antibodies that are specific for the spike protein, we looked at their gene usage, and it turned out that about 90%, almost 90% were IgG1. There were a few IgA and IgM. In terms of comparing these antibodies to normal, to healthy individuals, we saw an excess usage of V3-30 in the heavy chain, and the kappa was kappa V3-20. And in terms of J usage, there was an increased usage of J6, which led to a longer CDR3 length. Interestingly, the somatic hypermutation level was exceedingly low at 2 or 3%. This actually suggests that one could get pretty good antibodies directed to the spike with minimal affinity maturation, just largely relying on VDJ recombination and heavy light chain pairing. In some ways, this bodes well for vaccine development. So naturally, we looked at the 5 cases and all the antibodies and picked out the ones that are most potent in neutralizing the virus, shown in the color lines. And we found 18 that were most potent. This one was missed in the initial screen, but picked up later in the subsequent characterization. So in total, there were 19 pretty potent antibodies that were characterized in some detail. And first, we looked at these 19 antibodies for their binding profile to the trimer, to the RBD region, and to the N-terminal domain, or NTD. And you could see for the antibodies listed on the top panels, we saw good trimer binding for all of these. And these obviously recognize RBD, but not NTD, except for antibody 230, which we know to be a bit sticky and binds to a lot of different proteins at high concentrations. And then for this middle set of antibodies, you could see they all bound the trimer and bound NTD at variable levels, but not to RBD. And even though these were relatively low here at the bottom, as you'll see later, they are truly NTD directed. Interestingly, we have 2 antibodies, 243 and 251. The latter bound the trimer just fine, but did not recognize RBD or NTD. Interestingly, 243, which we fished out using the trimer, did not bind the trimer by ELISA and did not recognize RBD or NTD. And I'll come back to talk about these two because there are some interest. In terms of their neutralization profile against the pseudovirus shown on the top and against the authentic virus shown on the bottom. And you could see for the RBD-directed antibodies, we have a few that had essentially IC50 of 10 nanograms or less in the pseudovirus assay. And against the live virus, we had 4 that were under, with IC50 under 10 nanograms per ml. So these are extremely potent, with 215 consistently showing the greatest neutralization potency. But we also got a collection of NTD antibodies, and you could see that they're in general less potent than RBD-directed antibodies with slightly higher IC50 numbers. And then for these antibodies, we often see a plateauing effect that is at levels short of 100%. And this is reminiscent of some of the glycan-dependent antibodies in the HIV field, but we're not sure if the explanation applies here. And then moving down to the live virus, you could see 3 of the antibodies also have IC50 less than 10 nanograms per ml. So quite potent. In fact, more potent in the live virus assay than in the pseudovirus assay. And then the other 2 whose epitope could not be mapped up to this point had modest potency in the pseudovirus assay, but had pretty good potency in the authentic virus assay with IC50s, again, under 10 nanograms per ml. So these were the 19 top neutralizers that we had found. And of course, we wanted to understand their epitope. And so we went on to do epitope mapping. But before doing that, we wanted to throw in a number of weakly or non-neutralizing monoclonals to make the effort more complete. And so we took these additional monoclonals from different cases, color-coded as before. And then these obviously are non-neutralizing, and these are very, very weakly neutralizing. And we know some of these are directed to RBD and some not. And so these were tossed in with the previous 19 to do competition ELISA to understand their competition profile. And this was done in 2 batches, one for non-RBD binders and one for RBD binders. And it was done in a checkerboard fashion, and the results are presented in this heat map. And what we've done was to take one set of monoclonals and biodetonate each, and then use the same set to compete. And immediately you could see that there is one large cluster, which we call cluster A, that's partially overlapping with cluster B, and then there's a cluster C that is non-overlapping, and there's a unique antibody that does not compete with any other. All of these antibodies bound the trimer by ELISA nicely. And then on the bottom here, we had the binding of these antibodies to NTD. And you could see this large cluster is essentially all NTD directed, except for one, and that's the 251 that we could not map. But obviously, it falls within this region. So we suspect it's recognizing a quaternary epitope in the NTD region. And then these 2, one is overlapping with a larger cluster, and this one is non-overlapping. Each as you could see here, each has a member that bound NTD. So even though they're distinct clusters, they are in the NTD region. On the RBD side, we see, once again, one large cluster that's linked to a second cluster, which in turn is linked to a third cluster. And then that last cluster here is also overlapping with the third. And so now coming to the bottom here, if you look at competition for ACE2 binding, this large cluster is essentially knocking out ACE2 binding except for these two. And also, if you see that we could make certain mutations in the RBD that destroy ACE2 binding, and many of the monoclonals would lose binding when tested with this mutant RBD. So this all suggests that this large cluster is directly interfering with ACE2 binding. And then that's linked to another cluster. And then the third cluster here has a monoclonal, a known reported monoclonal called CR3022, which has been mapped to the side of RBD when the RBD is in the up position. And so this helps us to localize the third cluster, which we call cluster G, to the side of the RBD. And then this fourth cluster, but interestingly, fourth cluster has one member that competes with something in the first cluster. So this likely bends back, and we'll map this out in a minute. So these could be done for all the monoclonals that bound the trimer by ELISA. But as I mentioned, we have one member, and that was 2-43, that did not bind the trimer in ELISA. However, it did bind the trimer when expressed on the cell surface. And as you see in this panel here, it bound the trimer well, but that binding cannot be competed out by RBD, NTD, ACE2, or the trimer, but it's competed out by itself, of course. Then in terms of competition by NTD-directed monoclonals, there's either no effect or very minor effect, but they are largely competed out by RBD-directed antibodies. So we surmise that it's likely recognizing something in the RBD region, but maybe quaternary in nature, and perhaps it's slightly fringing on the NTD as well. And as you'll see later, this speculation panned out when we had the structure. So in terms of the epitope mapping results as represented by the Venn diagram, we present the RTD region here. And in this case, the potent neutralizers are shown in the dark red. The weaker, relatively speaking, weaker antibodies are shown in the pink. The non-neutralizing antibodies are shown in black. And this orange circle represent all those antibodies that bound NTD. So as you could see, the cluster A, the large cluster A is actually these three overlapping diagrams. And then cluster B is this one, which has a member that clearly falls within the NTD binding profile. And then cluster C likewise has one member in here. And then this unique antibody 1-21. On the RBD side, we once again, the color coding is the same as on the left. And so we have the potent antibodies mapping largely within this green circle, which represents competition with ACE2. And then this yellow oval is the competition results presented by cell surface staining. So that monoclonal 2-43 clearly competes with these antibodies, but does not bind RBD. But it must be in the RBD neighborhood. Then we have the next cluster here, which is partially overlapping with the major cluster here. And then this in turn overlaps with this cluster G represented by CR3022. So this is on the side of RBD, and then the final cluster actually loops back. So this must be the top of RBD. This is the one side of the RBD and coming to another side of RBD, making its way up to the top once again. So we next worked with Larry Shapiro in biochemistry and his student Mika Rapp to determine the Cryo-EM structure of several of the antibodies of interest. The first one done was 2-4, which is an RBD-directed monoclonal. And this is now solved down to about 3.5 angstrom resolution. And you could see the FV version of the antibody shown with the heavy chain in dark blue and the light chain in cyan. Essentially keeping the RBDs all in a down position. And obviously, it's engaging the RBD exactly the way ACE2 is engaging RBD. So such antibodies, like 2-4, is clearly neutralizing as a competitive inhibitor. We also looked at an NTD-directed monoclonal 4-8, and here in a lower resolution Cryo-EM structure, you see it is indeed coming at NTD at again from the top, but it's at a slight angle. And with the side view here, it's clearly touching the tip of NTD without affecting RBD. So the mechanism of virus neutralization of such NTD-directed monoclonal is unclear to us, and is certainly an area for further study. Now for that antibody of interest, 2-43, which we could not map, but we just recently got this structure, again, low resolution. I think in a few more days, we should have a higher resolution structure for this. But at this point, we could see that it is binding in the crevice between RBD and NTD, with some of it touching RBD and a little bit touching the NTD, hence pretty much consistent with the competition results. And this is simply the top view of the same thing. So taking all the information we have, we try to sort of write on here the potent neutralizing antibodies, the weaker antibodies in pink. And you could see essentially most of the potent ones are directed to the top of the spike, and the weaker ones generally recognize the side. And this is top view shows pretty much the same thing. This is in fact RBD here. This is NTD here. And those that come at the top, ACE2 will bind pretty much this surface. And those potent ones generally are competing with ACE2. And NTD, likewise, the potent ones are coming from the top, and the weaker ones are directed to the side. So far, all the neutralizing antibodies we have isolated and characterized are directed to the top of the molecule, suggesting that both RBD and NTD are quite immunogenic. And obviously, vaccine efforts that focus on RBD alone will miss one important class of the antibodies, i.e. the NTD antibodies. We have yet to find anything directed to the stem, and maybe that's because of our way of screening, or maybe that's a real phenomenon. We obviously are putting a couple of these antibodies into clinical development as potential therapeutic or prophylactic, and we have been comparing our antibodies in a landscape analysis with those that are uh, uh, in development or possibly going into development. And as you could see here, for RBD-directed antibodies, ours shown in red compare quite favorably with those that are developed by Regeneron and are already going into the clinic, as well as antibodies by other groups. And my former colleagues from Rockefeller have done a great job of isolating RBD-directed antibodies as well. And then what's unique for us is we have isolated antibodies that we think are directed to quaternary epitopes, as well as NTD antibodies. In general, they're slightly less potent than RBD-directed antibodies, but they are plenty potent. And so this is something unique up to this point. In terms, others have found NTD-directed antibodies, but they're generally very weak. We obviously have been looking at combinations as well, and I just show you one example of a particularly good combination that combines the most potent antibody 215 with one or the other NTD-directed antibody. And in general, compared to the 215 alone, there's not that much change in the IC50, but the IC90 could be shifted substantially to the left. And essentially, we could achieve 90% neutralization at about 10 nanograms per ml. So that's fairly impressive. So we just got results back from our collaborators at University of Hong Kong, where they took our most potent antibody 215, injected into hamsters on day -1, and on day 0 they did intranasal virus inoculation, and on day 4 they sacrificed the animal to obtain lung tissue and did RT-PCR to quantify viral RNA using primers directed to the polymerase, as well as beta-actin for normalization. And then when these results are expressed, you could see a control antibody. In this case, we use an HIV control antibody. Here are the viral levels in the lung tissue. And then for 215, we picked a dose that's actually quite low. Most people doing such experiments are using 10 to 20 mg per kg. We used 1.5 mg per kg and got 3 to 4 log reduction in viral load. And then for the low dose, we actually intentionally picked something that we expected to be only partially effective. And in this case, our guess was pretty spot on, and we see a very modest reduction. So we will be carrying out many more such experiments in the hamster model and other models. So I will finish up by telling you who did the work. The patients were organized by Michael Ng, who's in the infectious disease division. And then the folks at Aaron Diamond who did the work, Li Hong did the B-cell sorting, 10x genomics, and subsequently the bioinformatics analysis was done by Zizang Shen, and then antibody construction made by Jian Yu. And then the competition studies were done by Li Hong and Chen. And then antibodies were passed on to do pseudovirus neutralization by Pengfei Wang, as well as live virus neutralization by Manoj Nair and Yaoxin Huang. I already mentioned that Mika Rapp and Larry Shapiro did the Cryo-EM, and Yang and Neil did the project management. So I will stop there. Thank you very much for the opportunity to speak again.
All right, David, thank you very much. Very exciting. Now, these antibodies are clearly going to be important. They're going to be therapeutically useful and incredible reagents to look at spike. So I could start off with questions. People who want to ask should raise your hands, and Jesse will unmute you, but let me start. So it's to me particularly interesting that you're getting essentially germline responsiveness in your patients that are doing so well at making antibodies. I wonder to what extent, therefore, the variation in the repertoire in people to people it will be a big determinant in their ability to respond. And I wonder how universal it will be that people have germline, effective regions.
In looking at the other people's publications on this subject, it's pretty clear that generally this is not like HIV where there's so much somatic hypermutation in the potent broadly neutralizing antibodies. We only study 5 cases extensively, so it's really hard for us to speak about repertoire among different individuals. But even among the 5 cases, I mentioned there's 1 case that did not make any RBD-directed antibodies. And then I didn't mention this, but patient 2, who is a 71-year-old Hispanic female, was our champion in that she gave us 9 out of the 19 potent antibodies. It's amazing that, and if we didn't hit on that particular case, we would have lost about half of our collection of good antibodies. So there's
I guess all that means there are many people who have very low response. I mean, in fact, many people mount very weak responses, and maybe that's a germline repertoire issue for them. In fact, you have to wonder, all the people that have super low antibody responses, how did they clear the virus as many did? And are they using T cells or what?
Yeah, there are many other components of our immune system. So they must be doing a pretty good job, because a lot of the people who clear the virus in a matter of days and don't suffer much, they don't have a very robust antibody response in our hands. And I suspect Paul and others will speak to that as well.
It's another small question is, it's interesting that there's pretty good correlation of the reactivity to pseudovirus and real virus, but sometimes discrepancies. So I guess that's saying that sometimes the presentation of the spike is a little different between those 2 settings sometimes.
It's clearly different, but it's not different in a predictable way. We see that for some antibodies, pseudovirus gave a better result, and for other better results in terms of greater potency. And then for others, the authentic virus would give a more potent result. So it's different in ways that we don't see a pattern at this point.
Okay, let's throw it open to a couple if there's any more questions, and then we'll only have time for a couple. Jesse, any?
Jahar, you can speak now. I unmuted you.
Right. Yeah, right. So David, thanks for yet another exciting presentation. My question is about the last experiment you presented in rodents, where you gave the antibody and then instilled virus in the lung and showed that the viral load was reduced, if I got the experiment right. My question is, I'm curious as to in which compartment this viral neutralization is happening. Is it the airway compartment or the blood compartment? And when you do PCR on lung tissue, I suppose it's difficult to distinguish between the two, but I was wondering if you've done any control, say, on blood or on other organs to work out this particular question, because surely to neutralize airway virus, you need the antibody to reach the airway lumen. And I've been perplexed about this issue.
Well, Johar, it's a very important question you asked, but I don't have any answer for you because this is a first pass at in vivo experiment with these antibodies. We simply took, the colleagues in University of Hong Kong simply took the lung tissue of these hamsters and ground it up and measure for RNA for virus and beta actin. In terms of, they're doing some histopathology now, but we won't have those results for another week or so.
Sorry, just to follow up very quickly. If you're getting antibodies in the blood, surely the virus is escaping. Surely the virus is escaping the lung somehow, and producing the antibodies outside the airway compartment. And this is why I was thinking that the neutralization may be happening in an extrapulmonary location, namely outside the airway, extra airway location. But sure, I understand that these are very early experiments at this point. But perhaps a blood neutralization study would sort of get at this question.
Yeah, well, Johar, we talked about labeling antibodies and giving them to you, so you could try to answer that question more directly in a model.
I think we could have time for one more, and then we better move on. Who's next?
I think Sumit is next, one of the panelists.
Yeah. Yeah. Can you hear me?
Yes.
Yeah. I had two quick questions. One, for the hamster study, did you observe rescue in weight loss at those reductions in titer? And second, unrelated questions, do you see any evidence for neutralizing antibodies that don't target spike?
For the second question, it's not something we have looked at, so I'm not able to answer at all. For the hamster question, there is, I'm told that there is minimal weight loss in this particular experiment.